There's nothing that they can do. They are helpless. It's the people versus the scientific world. And it has surfaced, and the people are now speaking out.
I am convinced that when the people of the community understand the unbelievably unknown nature of the dangers, tampering with millions of years of evolution, they'll say no.
The city council should not play with it. You should not ban it completely forever. That would be a very, very stupid thing to do.
Cambridge, Massachusetts, June 1976. A busy, crowded city and home of Harvard University. The city council is about to take action that will stun the world of science. It asks Harvard not to begin a new type of research until the council has decided whether or not to ban it completely. The research planned for this laboratory involves the engineering of genes.
Once upon a time ago.
In Cambridge, the debate over the new experiments has spilled into the streets.
We, Science for the People, are here to try to bring the issues of this controversy to the public, to the people, because the people are at risk and are at benefit from the experiments that are being done with recombinant DNA.
The idea of genetic engineering going on in his city horrifies Cambridge Mayor Alfred Vellucci.
I'm worried about all the presentations that were made by scientists at the city council meeting. It was stated in the city council that anything could come out. In fact, they don't even know what's going to eventually come out of this experimentation. It could be anything. It could be contamination, infections, something that could crawl out of the laboratory, such as a Frankenstein.
The feeling has been generated that we want to work in a laboratory with things known to be dangerous. That's not at all the case. Nothing we plan to work with is known to be dangerous. Now, as to the unknown, one can do one's best about trying to figure out what might lie ahead, but research deals with the unknown.
This film will show how scientists and the public are trying to come to terms with a dramatic new technique, a technique that gives scientists unprecedented power to manipulate nature. But it was devised to help understand nature, to unlock a secret that has remained inviolate since life began. How a simple acorn becomes a mighty oak. A community of specialized cells, trunk cells, bark cells, cells forming branches, twigs, stems, leaves. How a human being inheriting a set of genetic instructions from its father and mother can develop from a single egg cell into a baby. Every cell of the billions that make up this 6-week-old girl contains the same set of genes. Yet in the process of development before birth, her cells went their separate ways. Somehow different genes were switched on in the different cells, making them skin cells, nail cells, cells of eyes, mouth, and hair. How these genetic switches work in a creature as complex as a human being is a total mystery. Perhaps the most intriguing mystery facing scientists today. The genes of this creature are better understood. A bacterium that lives in the human gut. It's called Escherichia coli, E. coli for short. It's the most studied organism on Earth, in part because it's so cooperative. A single E. coli in a drop of water will multiply itself enormously if given a few simple chemicals to eat and if it's kept warm and gently shaken. In a day, the original bacterium can become 100 billion. E. coli also gives up its genes very easily. Detergent bursts open the microbes, spilling out their genetic material. A little alcohol allows the genes to be spun onto a glass rod. This is deoxyribonucleic acid, DNA. It's the material not only of E. coli's genes, for the genes of all living creatures, including, of course, the genes of man. An E. coli bacterium ruptured with detergent lies with its DNA strewn around it. But also in the photograph, magnified 100,000 times, is a tiny loop of extra DNA called a plasmid. This plasmid is the key to the new technology of gene transplantation. A centrifuge begins the process of harvesting plasmids. Its rotor has been spun at high speed for 24 hours. In it are tubes containing the DNA from ruptured E. coli. The huge forces generated by the centrifuge have separated the plasmid DNA from the rest of the DNA in the bacteria. Under ultraviolet light, a dye makes the two separated bands of DNA glow visibly,